Hello, my name is Abraham Waldman. I am a senior honors chemical engineering student with a concentration in biochemical engineering. For my thesis, I'm doing research within Professor Todd Emmerich's research group in the Department of Polymer Science Engineering on extending droplet reach with polymers. A little bit of background on this project. Smart droplets have previously been used in surface cleaning and repair. So if you can imagine a polymer film with the surface chemically modified, if a tensile force is applied along the horizontal axis of this film to crack it, scattering nanoparticles from within the film on the surface of it, you can then flow smart droplets over the surface to pick up the droplets, cleaning the surface, and then to deposit the droplets within or back into the damaged substrate, helping to restore some of the mechanical strength and thereby repairing the substrate. The purpose of this study is to examine the extension of droplet capability by the addition of polymer arms. And so this allows us to explore fundamental behaviors of droplets, explore the attachment of arms at fluid-fluid interfaces between two miscible fluids, shown here in figure 1, where fluid 1 is immiscible in fluid 2, and the polymer arms are attached to fluid 1, encapsulating it, and then extending into fluid 2, and to develop an understanding of the interactions of fluid interfaces between two immiscible fluids. This has applications in using droplet arms for recognition, transport, and capture. And for this research, I'm working directly with postdoctoral fellow Dr. Zifei Yang within Professor Todd Emmerich's research group. Previously, within Professor Emmerich's research group, Dylan Barber et al. Published a paper entitled Programmed Wrapping and Assembly of Droplets with Mesoscale Polymers in 2020. Mesoscale polymers are essentially polymer-based ribbons, which have the ability to modulate interactions with liquid droplets. Specifically, they've been used within this paper at the interface of oil and water droplets. And what this paper did is it had two different types of polymers prepared using flow coating. The first was a pH-responsive polymer, polydimethyl amino ethyl methacrylate. And the second was a polymer that had both hydrophilic and hydrophobic regions called a diblock copolymer. For the purpose of, or for this polymer, it was poly-T-butyl methacrylate with trace benzophenone and fluorescent. Shown here in figure 2 is the pH-responsive component of the first polymer that I mentioned, in which at an increasing pH, the amine groups deprotonate, and they therefore become more hydrophobic and are more easily able to interact with hydrophobic fluids, such as perfluorodecyl oil. And so shown here at a low pH, when these amine groups are not deprotonated, they extend, the polymer ribbon extends in solution, but does not physically wrap the droplet. At a pH of 8, when some of the amine groups in the polymer backbone are deprotonated, we start to see some wrapping of the oil droplet, but we still see a good deal of extension in the solution. And at a pH of 10, we see the entirety of the oil droplet wrapped in deprotonated amine groups. The second polymer ribbons that were made in this paper were mesoscale block copolymer ribbons. And what's happening here is that 90% by mass of the polymer backbone is made up of tert butyl methacrylate, shown here in this red box, which is a highly hydrophobic component. And this can be deprotected to form carboxylic acid group, shown here in this red box, which is a highly hydrophilic component. And so the hydrophobic component will wrap around the hydrophobic fluid 1, shown in this wrapping of these figures, or excuse me, of this droplet. And the hydrophilic component will extend into the hydrophilic fluid, where hydrophobic fluid 1 is immiscible in the hydrophilic fluid 2, thereby giving polymer arms that are able to both wrap the droplet and extend the solution. However, there were two drawbacks to this paper. The first one was that the polymers were prepared using flow coating, which is not a scalable process. And the second was that this only studied interaction with a single droplet. And so the purpose of my thesis, which I'm working on with Dr. Zifei Yang, is to study the extension of droplet reach with polymers for multiple droplets using photolithography and spin coating for ribbon preparation instead of flow coating, which allows us to scale up at a much larger degree. And so the polymers are synthesized using raft polymerization, which is a controlled radical polymerization process. And the first polymer that we were working with contained cross-linkable and fluorescent monomers, allowing for the manipulation of mechanical properties via the cross-linkable component and microscopic tracking via the fluorescent component. So prior, as I previously mentioned, prior ribbon making using flow coating limited in its scalability, and so we are using photolithography, which is typically used in the synthesis of computer chips to scale this process up. And so, for an example of what I'm talking about, we start by depositing or spin coating polystyrene sulfonate onto a silicon wafer, or PSS. And the premise of this is that PSS is easily miscible in water, and therefore, by placing this in water, we can release the ribbons into solution. And then the uncross-linked polymer is spin-coated on top of the PSS layer. Benzophenone cross-links at 365 nanometers under a photomask. And so where these red boxes indicate the photomask, the areas directly beneath them would not be cross-linked, and the green areas, which are exposed to the 365 nanometer wavelengths, would be cross-linked. The uncross-linked polymer is soluble in chloroform, but the cross-linked polymer is not. And so by rinsing the chloroform, we can remove the uncross-linked component from the structure and be left with these polymer ribbons, which are all hydrophobic. The next step is to transform these ribbons from hydrophobic to hydrophilic, or parts of these ribbons. Again, because the hydrophobic component would be wrapping around the oil droplets, and the hydrophilic component would be extending in the solution. And so the polymer that we're using is the same polymer discussed in the previous paper, polybutyl methacrylate with trace benzophenone shown on the left and fluorescent, where benzophenone is used for cross-linking and cross-links at 365 nanometers, and fluorescent is used for optical microscopy. This torpedo group can be deprotected using 254 nanometer wavelengths when a photoacid generator, or PAG, is mixed in with the polymer, which causes this torpedo group to deprotect, shifting the torpedo methacrylate group carboxylic acid, and thereby shifting this polymer from hydrophobic to hydrophilic. And so if you imagine that after 365 nanometers, we have a totally hydrophobic component where this entire ribbon would be dark blue, we are then able to cross-link, or excuse me, to irradiate at 254 nanometers part of these ribbons to turn some part of them into hydrophilic ribbon, and therefore making diblock polymers that are partly hydrophobic and partly hydrophilic. And so shown here are ribbons before release into water, the hydrophobic, fully hydrophobic ribbons release into water, and fully hydrophilic ribbons after release into water. And again, we're using water here because it dissolves PSS, making it easy to release these ribbons in solution. However, one problem that we were seeing with this polymer was that we were seeing leaching of the fluorescence in the solution. And so we decided to try a new polymer, coumarin methacrylate, torpedo methacrylate, copolymer of those two monomers, because coumarin, which is the cross-linker group, is also fluorescence, and so the fluorescence group can be completely removed from the polymer, so it would no longer be leaching it. And so this is the coumarin methacrylate, torpedo methacrylate copolymer. Again, torpedo methacrylate is used because after a deprotection using PAG at 254 nanometers, it's able to change the deprotected part of the polymer from hydrophilic to hydrophilic. And coumarin cross-links at 365 nanometers to form this cross-linked version of coumarin. And in doing so is also fluorescent and is therefore able to be visualized under optical microscopy. However, the 254 nanometers used to activate the PAG decross-links the coumarin, and so another step needs to be added in of a recross-linking at 365 nanometers to recross-link the coumarin. The rinsing step after the first cross-linking step has been shown, however, to remove some PAG, and so what we're currently working on at this point in my thesis is the potential of adding PAG directly into the monomer as a third methacrylate group, directly into the copolymer as a third methacrylate group, and methacrylate PAG monomer, which would therefore make it so that the PAG is completely dispersed in solution, or within the polymer, and fully deprotects when irradiated at 254 nanometers. Another option would be to spin coat the PAG on top of the uncross-linked polymer or to increase the concentration of PAG present mixed in. Polymer characteristics are being quantified using ellipsometry, which is able to measure the thickness of polymer films, optical profilometry, which is able to measure the dimensions of polymer ribbons, and pendant drop tensiometry or contact angle measurements, which provide us with an understanding of the degree of hydrophobicity or hydrophilicity of the protected and deprotected groups, respectively. We're currently able to create 90 nanometer thick ribbons that are able to successfully wrap multiple droplets, but at this time show no evidence of the hydrophilic component extending into solution, which we hypothesize is due to PAG being rinsed off. And so we're seeing that the PAG is rinsed away, or rather that since the PAG is rinsed away, there is not as much of a hydrophilic component, so it does not extend into solution. And we're also seeing that that the ribbons are rather fragile and break apart in solutions. So as you can see on this rightmost image, this ribbon is not very long. And so this may be because of low ribbon thickness or because of the decross-linking component that occurs at 254 nanometers, which is what we are currently working on at this stage of my thesis. I'd now like to acknowledge and thank my committee chair, Professor Todd Emmerich of the Department of Polymer Science, my second committee member, Professor Jessica Schiffman, the Associate Department Head of the Department of Chemical Engineering, Dr. Zifei Yang, the postdoctoral fellow within Professor Emmerich's research group with whom I'm working on this project, the College of Engineering, specifically the Department of Chemical Engineering, the Department of Polymer Science and Engineering, the Commonwealth Honors College, and the UMass Undergraduate Research Conference. The funding for this project was provided by the Department of Energy with grant number DE-SC0008876. Thank you.